Here we're looking at cell respiration. So our first stage occurs outside the mitochondrion in the cytoplasm of the cell. And our first step is glycolysis, which means sugar splitting. So we start with some glucose, which enters in here. The first stage is that it gets phosphorylated. That means phosphorus or phosphate groups are added to the sugar to destabilize it. So we need to include 2 ATP that actually gets used in this stage. So there's an input of ATP, even though at the end we're going to get more out of it. So we've phosphorylated our sugar, which means it can be split, which is called lysis. You also see that root of that word in the organelle lysosomes, which contain digestive enzymes, or hydrolysis, splitting and adding water. Now, the next stage, which is really important for later on, is that we actually charge or reduce carrier molecules. So we're going to put 2 NAD plus here. Now, these are actually called coenzymes, and they are reduced, which means they gain electrons and hydrogens in this case. So we can think of it as being a carrier, a little bus for our hydrogens and electrons. So that becomes NADH and H+. So there's a hydrogen and its associated electron here. It's part of the molecule, and an extra hydrogen ion that gets transported with it. Now what goes, a transfer of electrons in a chemical reaction, if we have a gain of electrons somewhere, we must have a loss of them somewhere else, because we can't magically get electrons out of nowhere. So we also have oxidation of our sugar at the same time. So we've taken our glucose, we've used some energy and some phosphorus to phosphorylate it, we've split it, we've oxidized it, and in the process we've reduced some carrier molecules. Now we also, we actually get 4 ATP out of this process. So we used 2 and we got 4, so we have a net gain of 2. That also means we must have used some ADP over here. And that's freely available in the cytoplasm, because for all the ATP we generate, that's getting used up by cellular processes and recycled, those low energy components, back to ADP. Now, this part here, our charged or reduced carrier molecules, NADH and H+, they're going to be important at the end in what we call the electron transport chain. So we'll pop that there. Now here, pyruvate, that's the molecule we get that's left over from the sugar. Now we actually have 2 of those for every glucose molecule. Now this enters into the stroma, the matrix. I'm getting confused now with photosynthesis. The matrix is the liquid part inside our mitochondrion. So let's just label that for a moment. So here, this box here will say is our matrix. Whereas this here, these infoldings, so our mitochondrion is a double membrane bound organelle, which is evidence for endosymbiotic theory, that an organism like a prokaryote, here our mitochondrion was engulfed by a larger cell, so the existing membrane from the prokaryotic cell, or the smaller one, remained, and the amoeboid action of the cell surrounding it, that membrane was also retained. We also see in mitochondria that they have their own DNA and ribosomes for manufacturing their own proteins, and that the size of the ribosomes is 70S, just like prokaryotes today, so that gives us evidence for that theory. Now here we would call these infoldings cristae, so we label these, and we can also label that inside one crystal we have the into membrane space. And my writing is a bit big for that, sorry. Okay, so entering into the mitochondrion, into the matrix, the liquid component, we have the link reaction. Now, of course, that's happening twice for every, we have each pyruvate molecule. We'll factor that in times 2 for each glucose molecule that we started with. So in the link reaction, we need to take our pyruvate and we need to decarboxylate it, which means we remove CO2. So carbon dioxide leaves. Now that's 1 for every pyruvate, or 2 pyruvate molecules, 1 glucose. So carbon dioxide leaves and that will diffuse out. Now what's left over, our 2 carbon molecule, needs to be attached to a coenzyme. So coenzyme, it's called coenzyme A. That transports the 2 carbon molecule. Okay, so together, the coenzyme and the 2 carbon group, the ethyl or acetyl group here, so 2 acetyl-CoA, that gets taken to the next stage, which is called the Krebs cycle. And I can't write today, apologies. Okay, so we have that, and of course if we're using that coenzyme, we should indicate that that enters the link reaction and acetyl-CoA. Okay, also at this stage in the link reaction, as well as decarboxylating the pyruvate, we also get more of our carrier molecule, our NAD. So for 2 of the link reaction, we get 2 NADH and H+. So we should really pop actually 2 NAD here as well, that's going in. Okay, so heading into our Krebs cycle now. Our 4 carbon molecule that's already existing in our Krebs cycle is going to join, perhaps if we showed a continuation of the arrow from here, our acetyl-CoA. So our 2 carbon component is going to join the 4 carbon molecule in the Krebs cycle to make a 6 carbon molecule. And then the coenzyme, CoA, is going to come off again straight away. So we can pop perhaps just 2 CoA leaving the cycle again, so that can be reused in the link reaction. So our 6 carbon molecule here, now we decarboxylate that. So we lose a carbon dioxide to make a 5 carbon molecule. So carbon dioxide comes off at this stage. So we have decarboxylation. And then another one again. Carbon dioxide here. So for 1 turn of the Krebs cycle, we're losing 2 carbon dioxides. But again, if we're thinking for each glucose molecule, 2 link reactions, 2 Krebs cycles, we would say 4 CO2 in total. Now we're also getting more carrier molecules, more NADH. So we can draw that as an arrow with an input. NAD plus is coming out as NADH and H plus. So we're losing some hydrogen. There are also some reactions involving water in the Krebs cycle which aren't shown here. That's where some of that's coming from as well. And another one here. Oh, I'm drawing too many pluses, sorry. All right, and this last stage here, we've got 4 carbon molecule staying as a 4 carbon molecule, but it's going from a much higher energy arrangement here to here. And we're regenerating our original molecule so that we can continue around the cycle. So we have some ATP gained at this point. So we'll put ADP. an inorganic phosphate, we get a little bit of extra energy generation. We get another, so it's going to get a little squeezy there, another NAD plus becoming NADH and H plus. And there's one last carrier molecule that we haven't seen yet, an extra one that exists in the Krebs cycle called FAD. Very, very structurally similar to NAD, so just another type of bus ferrohydrogens. But both are associated with that molecule, so we write FADH2. Okay, so this last step's quite big, a lot going on here. So we've had decarboxylation, lost some carbon dioxide. Our carbon itself is getting oxidized. So oxidation of carbon, which is paired with the reduction of carriers, just like in glycolysis. And of course, we've